So keep in mind that we just talked about the cell-to-cell contact dependent. So those would be the cell adhesions that we see, and the cell-to-matrix contact dependent communication, the hemidesmosomes as an example there. We also talked about receptors that bind ligands, and so we can classify these different receptors. We can put them into little groups. Okay. In addition to the transmembrane receptors, these proteins that are found, well, let's sketch it out. Why not, right? So if we imagine then for ourselves, we're going to have these receptors. Let's have this be a cell membrane right here. And if we have, let's go ahead and put another cell membrane out here. Which would make this my interstitial fluid. And this would be my intracellular fluid on the cell on the top, and this is my intracellular fluid on the cell on the bottom here. So speaking of cell contact proteins, then we would have, of course, the communication from cell to cell, as we discussed, with these proteins connecting with one another in the extracellular environment. And so these trans proteins crossing over, connecting to one another, and then we would, of course, have our signaling cascade of cells that exist, of ions that exist. Maybe it's time for me to go to bed. Enzymes or other protein signaling molecules on the inside that then would communicate that we have this cell-to-cell contact being occurred. So this would be my contact dependent. Signaling. And as we discussed, that can be temporary or long-lasting depending on what the nature of that contact is. We can have cell-to-cell. We can also have cell-to-matrix, which I'm not going to draw right now. And so those would both fall into the category of these contact-dependent communication. And we could, in fact, call these my receptors. Okay, those are my receptors. And what's interesting is they both act as ligands. So the ligand for the receptors that are down here would in fact be the receptors that are on the other cell. So we could say, you know, if these are acting as receptors, these are the receptors ligands, and vice versa. And so it is important to understand that relationship then between these proteins. We'll get into that in more detail. Then we have transmembrane receptors that are hanging out here, and so I'll just draw this one here, and we're going to have a extracellular binding site. And that, of course, is going to be in the extracellular fluid or the interstitial fluid, and that's where your ligand is going to bind to stimulate a response on the inside. And we would have proteins. Again, we're going to have this signaling cascade of these proteins on the inside that are going to be activated one right after another. Okay, so you know, this activates this one, and this activates this one, and so on and so forth. Same here. The receptor activates this one, which activates this one, which activates this one. Okay, and so we would call this a ligand binding. So ligand dependent signaling. But there's one more type of receptor that I need to emphasize, and that receptor would actually be found in the cytoplasm or sometimes in the nucleus. Okay, so we could throw in a nucleus here, and we could have a receptor that would be hanging out in here as well. already bound to the DNA. So this would be my DNA. In both of those cases, they're still ligand dependent. But the difference is, in this case, the ligand actually would enter into, cross my cell membrane into the cytoplasm to bind this. And so we would have my ligand and my receptor. My ligand and my receptor. Okay? So we can classify these receptors based on their location, on their function, and on the type of signaling molecules that they bind. If the binding site is in the extracellular fluid, then we know that the ligand is hydrophilic. Why do we know that? Well, because the extracellular fluid is mostly water, right? So if it's dissolved in the water, if it's floating in the water, then it's hydrophilic. Even these molecules over here, we don't need gigantic letters, so let me change that back down here. Even these molecules here, these proteins are in fact hydrophilic, or at least the regions that are exposed to water are hydrophilic. Okay, the regions that are exposed to the water are hydrophilic. So what about these receptors that can cross through the plasma membrane, or these ligands right here? Well, if they're getting across the plasma membrane by simple diffusion, which is what I was trying to indicate, then in this case, these are hydrophobic. Or another way of saying that is lipophilic. Okay? And so we're going to again classify these receptors based on location, but also based on their function. So I apologize, based on their, well, we can classify them based on their function, but based on their ligands that they bind. And so if we take a look at this, here's how we're going to look at this. Here's how we're going to classify this. Receptors that are located on the plasma membrane, they're transmembrane proteins almost exclusively. And that extracellular binding site is going to bind that hydrophilic ligand, as I talked about. Now, on very rare occasions, maybe I'll go down here and steal this. Yeah, this will work. On very rare occasions, the binding site for a transmembrane receptor is actually, so here's my lipid layer, phospholipids. And of course this region right here is hydrophobic, and we have a transmembrane protein with my extracellular portion here. Okay, so we're going to have that, and then we're going to have this protein cross this membrane. Okay, and we're going to have intracellular side that's going to interact with this intercellular portion, and then of course we're going to have more phospholipids. Well, in terms of this protein, these regions that I'm going to kind of scribble in that make them more purple than not, I guess I could highlight them purple, but anyway, these regions here would be hydrophilic because they're interacting with the water. But notice my region in the center here. This would actually be hydrophobic. And in some rare instances, the ligand will actually bind inside here. Okay, but that ligand then, if this is the case, this ligand would be actually hydrophobic. I'm sorry, that's so very messy. Let's write this outside here so you can actually read this. It would be a hydrophobic ligand, also called a lipophilic hydrophilic ligand. Okay, so that it would interact with those amino acids that are found inside that lipid layer. Okay, and so that's essentially what this is talking about here. Sometimes we've got ligand binding sites as being inside that fatty acid layer. It's still binding to a transmembrane protein. That's the key there. And then we've got our receptors that are located in the intracellular fluid or in the nucleus, and most of the time those are transcription factors. And so they will have multiple binding sites, and that's kind of a key there. They're going to have a binding site. I guess I have more room here. So let's say we're in the nucleus, or actually, yeah, we'll be in the nucleus today. So here's my nuclear membrane. And you'll remember it's a double membrane system. Membrane. Whoops. Okay. And my nuclear membrane, of course, houses my DNA. And if we were to take this DNA strand and open it up, so remember we're going to have my double helix strand like so, and we're going to open it up so that we've got our replication, or actually in this case a transcription bubble. Okay. I'm really hoping that this looks familiar to you. The intracellular receptors, okay, those intracellular receptors have multiple binding sites. They're going to have a binding site for the ligand, so we'll draw that there. And they've got a binding site for the DNA. Let's indicate that. So this is my ligand binding site. And then we've got my DNA binding site. We're still not finished. Remember me talking about transcription factors and what they do, and we talked about how they assemble other proteins. So we're going to assemble more transcription factors to that region. And so, you know, those transcription factors are going to be all recruited to this area. And each of these transcription factors are in turn going to help recruit ultimately my RNA polymerase. And you'll remember that we have RNA polymerase here. Okay. And so remember then this area right here, right underneath the RNA polymerase, this would be my promoter region. And recall that this is my regulatory region, right? Okay. And so you'll want to make sure you never forget that. This is important. Okay, this is really important. And as you go on to take genetic classes or molecular biology classes or so forth, you're going to learn more detail about that. But I want to make sure you at least have the basic kindergarten level gene regulation concept down. And so when I go back here to tell you that, all the way back up here, that they have multiple binding sites, that's what I mean. We've got a binding site for the ligand, we've got a binding site for the DNA, we've got the binding site to recruit these other proteins, and ultimately to recruit RNA polymerase. And so we've got these multiple binding sites there. And then also sometimes they will have a binding site. If we go back here, maybe we're going to have a binding site for a molecule like calcium. Maybe calcium's going to bind somewhere in here. Okay? Or sodium or something to that effect, magnesium, whatever you have. And so there would be another binding site for an ion. And so that's kind of interesting and important as well when we're looking at these molecules. Okay, these intracellular receptors. Now I'll emphasize not all of them are transcription factors. Give me an example of one that, or let me give you an example of one that's not. There's a protein involved in, it would help if there's a protein involved in skeletal muscle contraction called troponin, and it is not a transcription factor. but it does bind calcium. And that calcium will in turn, the net effect, it takes several more steps, but the net effect is that troponin calcium complex will initiate muscle contraction. And so troponin would be considered an intracellular receptor, and it's for calcium ion, but it's not a transcription factor. So, while many of them are, not all of them are. And so that, in a nutshell, tells us about the receptor classification. So I've created here a couple of flow charts, and I'm actually going to doctor this one up a little bit because I don't really like these labels right here. Actually, this one that I'm scribbling out is just fine. Whoops. Oh well. This is the one I really don't like. So let's rewrite them. This type of receptor, so we're dealing with transmembrane proteins here. Okay, so transmembrane proteins, these are the ones located in the cell membrane. Okay, and those cell membrane transmembrane proteins can be divided into two different groups. One group is those that are involved in cell-to-cell connections. These would be the contact dependent, or cell-to-matrix connections. So ultimately, these would be, the best way to describe these would be my contact dependent signaling. Okay, and while there are other different types of proteins involved in this process, including PCAM and selectins and other types of molecules, the one that we are going to be discussing is the integrin receptors. That's the one I'm going to use as our primary example of a receptor involved in this cell-to-cell or cell-to-matrix contact dependent process. Okay, when we get into the immune system, we're going to learn about some other ones. For example, something called MHC molecules, complex 1 and MHC complex 2, and we're going to learn about things like CD4 receptors and CD8 receptors. For right now, though, for this particular lecture, we're only going to talk about the integrin receptors. These will come later. Okay, we'll talk about those later. And then the other category, what I did is I messed it up because I just copied and pasted when I was making this flow chart here. So I got to rename this one. These would be my ligand dependent. Okay, so these are my ligand dependent, and if I were to make this a little prettier, I want a big one. We probably should just call this my contact dependent. Okay, to distinguish this, and so that's probably a better way of looking at it. Now for my ligand dependent, we can divide them further into what we're going to call ion channels. And I'm going to look at that and I'm going to show you those. These are ligand gated ion channels. Sometimes these are also referred to as channel receptors. Again, this tends to depend on the preference of the professor as well as the preference of the person writing the textbook, and so different textbooks will use different terminology. I most often use this one, which is why it's the one that made it into the box. I tend to call them ligand-gated ion channels mostly, almost exclusively. I very rarely say channel receptors or receptor channels. Then we also have another type that is going to have a dual nature, two different properties here. And these properties include a receptor and an enzyme. Okay, so what we typically see with this is that the receptor portion is in the extracellular fluid. Okay, so this would be the extracellular fluid facing portion of the protein, and then the enzyme properties are usually either in the membrane itself, so in that phospholipid area, the fatty acid area, or in the intracellular fluid portion. Now, Again, this is textbook specific, but these are sometimes called receptor enzymes or enzyme receptors, and sometimes they're called tyrosine kinase receptors, and that's actually my preferred term, is this tyrosine kinase receptor. Now, tyrosine kinase receptors are typically abbreviated. If I were to see this in perhaps a picture or a figure, we're going to see a couple different abbreviations for this. We're going to see TKR for tyrosine kinase receptors, but and it seems to be a little bit like United States versus European, but also it depends on where you trained and who trained who and so forth. So sometimes you see this tyrosine kinase receptors, and sometimes you see receptor tyrosine kinases. They mean exactly the same thing, okay? So don't get fooled, don't be fooled by what you see in the papers. If you were to go through the scientific literature, you could see either nomenclature used. And then last but not least, we have these lovely, lovely G protein coupled receptors. And this is the largest family of receptors, and we're going to look at these guys. There are so many of them, and they're a complicated type of receptor, and they also will have a portion that, in fact, what they are is they're not just, they're a receptor, but then a complex of proteins that assemble together with the receptor. And so you have a receptor portion. Let me go ahead and draw. Here's my cell membrane. And we're going to have a receptor that is going to have an extracellular binding region out here. So this would be my extracellular fluid, and most of the time, not all the time, but most of the time, this is my ligand binding site. Okay, remember, they're generally speaking, when we're dealing with receptors and molecules, there's always exceptions to the rule, and then we're going to have an intracellular binding site also. And that intracellular binding site is where this complex of receptors or molecules assemble, and we're going to have these assemble together, and I'm going to give you the details of that assembly later, but we'll have these different proteins that assemble together in the intracellular fluid, and we'll get into those identities and what they do and how they do it in a little bit. So there we go. That's my assembly. So we've got, that's why it's called coupled. Okay, so here's my receptor portion. Okay, that's my receptor. And that receptor is coupled to an internal intracellular assembly that we'll define that in more detail later. So that's how you classify your transmembrane receptors, transmembranes. And as I mentioned previously, these most often bind hydrophilic ligands. Most typically, that's what they bind. There are again exceptions to the rules, where sometimes the ligand might bind. I'll just use my G protein coupled receptor as an example. I might instead get a ligand binding there. Okay, in which case that would be binding in the fatty acid layer, and that would be an example of a hydrophobic ligand. But that tends to be a little bit more rare than the hydrophilic ligands. It still happens, and so I need to mention that it is possible, but it's more rare. And then if we go and look at these intracellular receptors, these beautiful things here, these ones, very important intracellular receptors. Okay, those have a tendency to be located in the cytoplasm. So here we've got our cytosolic or cytoplasmic receptors, and they are soluble proteins that dissolve in that cytosolic gel-like substance. and they would be located there, and those would fall under the category of transcription factors. If they're a transcription factor, when they bind their ligand, so when the cytosolic protein binds its ligand, it will migrate to the nucleus. But its default location is in the cytosol, okay? So that's its default location. And then there are other examples of proteins that are not transcription factors. Right here I've got a listed as a calmodulin protein, which is actually a calcium sensing protein. And we're going to see calmodulin show up when we deal with smooth muscle contraction. Calmodulin plays an important role in that. It also plays an important role in a number of other processes. It's involved in learning and memory formation within neurons. It's an important protein. And then on a previous area, where did I doodle my stuff? I don't remember where I put it, but it doesn't matter anymore. Going back here to this, the other example, remember, that I gave you is troponin. Which is ironically also a calcium sensing protein. So we could classify both of those. Calmodulin then would be involved in smooth muscle contraction, and troponin is involved in skeletal muscle contraction and in cardiac muscle contraction. So you're going to see troponin show up too. And so we get these different calcium sensing molecules. We're not just interested in sensing calcium. There's other molecules involved that will sense other types of signals, but calcium is a big one. It actually is very important in cell behavior. We use calcium as a signaling molecule on a frequent basis. Calcium is in fact a very common second messenger, and so keep that in mind. Calcium is a second messenger. And we'll see it playing a star role in the skeletal muscle, smooth muscle, cardiac muscle. We don't really we do talk about it in neurons. It plays a key role as a second messenger in neurons. And in many other situations, calcium is a very important second messenger. And then we have, and we're really not going to talk about them, I just want you to be aware of them. We do have plasma membrane receptors in organelles. I talked about just briefly, when we take our messenger RNA, you might remember I said we're going to add a cap to it. Okay, there's my cap. And part of that signal, that beginning signal, was a signaling sequence right there that really functions as like an address. And that signaling sequence tells the messenger RNA whether it needs to relocate to the endoplasmic reticulum or not. So it'll bind to the ribosome. Everything assembles together on the ribosome. Here's my ribosome. Okay. And if that signaling sequence says go to the ER, the whole thing will shift to the ER. And on the endoplasmic reticulum, there is a receptor for that signaling sequence, and so that receptor will recognize that sequence, and everything docks together, and then we begin to synthesize the protein on the endoplasmic reticulum. And that's really the extent of us talking about these plasma membrane receptors and other organelles. Just be aware that they're there, but we don't speak of them too much. Technically speaking, the electron transport chain could be considered a receptor in the mitochondria for electrons. And so, you can use the terminology in other situations. Now, going over here to the nuclear receptors. Nuclear receptors are almost exclusively transcription factors. I can't think of a single situation where they're not. That doesn't mean I'm wrong. I'm right. There may be the occasional nuclear receptor that is not a transcription factor, and if somebody thinks of one and knows of an example of one, by all means shout it out and let me know. Well, you can't shout it out in a video recording lecture because you're not here, but you know, just tell me later, because I'm interested to know of one. I don't obviously know all things. According to my 5-year-old, I don't know anything. She knows everything. So maybe I'll ask her about nuclear receptors. Anyway, I was pretty sure the know-it-all stage didn't show up until teenage years, but apparently I am wrong. Anyway, so that's the basics of receptor classification, and I want you to be able to understand this and maybe work on it together, work on it for you until you understand this association and classification. One more thing I need to add here is that these intracellular receptors, generally speaking, bind lipophilic molecules that can go through the plasma membrane by simple diffusion. Okay, simple diffusion. Oh, hemoglobin might be another example of a cytosolic receptor that hits this, although it's not really a receptor, it's just a carrier or binding molecule. But anyway, lipophilic molecules pass through the cell membrane, generally speaking, by simple diffusion. So, of course, remember, lipophilic also means hydrophobic. Again, exceptions exist. Calcium does not pass through the membrane, so calcium would have to go through a calcium channel, and so it would go through this channel through the membrane, and it might be going through the membrane of the cell membrane. We have that happen sometimes where calcium feeds in through the cell membrane from the extracellular fluid into the cell, but we also have frequently a situation where this membrane is not the extracellular fluid, but instead is the endoplasmic reticulum, and so calcium leaves the endoplasmic reticulum into the cytoplasm. So we'll have a chance to talk about those and be aware of those, but in that case calcium is not a lipophilic molecule. It is a hydrophobic it is not a lipophilic molecule, it is not a hydrophobic molecule, it is very much hydrophilic. And so again, we're talking generalities. Most of the time we're dealing with hydrophobic, but in the case of calcium, this guy's hydrophilic. Okay, and I think that is it on receptor classifications, and now we're going to go ahead and look at some specific types of receptors.